So yes, so I'm going to talk about subcellular organization of proteins. And as you know, cellular processes are partitioned in time and space. So knowing when and where a protein is present gives us good clues about its function. We also know that relocalization of proteins is an important form of functional regulation, and mislocalization can also be a cause of disease. So in my opinion, all systems level studies should include spatial proteomics or single cell proteomics data. So I'll try to convince you of that during my talk today. And as you heard in the introduction, I'm also co-director of the Human Protein Atlas. So it's a project that has been running for nearly 20 years now, and we're systematically mapping the human proteome using our in-house generated collection of antibodies and large-scale immunoassays such as immunohistochemistry and immunofluorescence. And this database has about, I don't know, over 1 million users on a yearly basis. So if you haven't used it, check it out. I'm sure there's something, hopefully something for everyone. But most importantly, the raw data that we produce is images. So we have over 20 million images that have been systematically generated and systematically annotated. So this is a great data set if you want to develop pattern recognition algorithms or machine learning models to interpret spatial information. And I'll get back to that later in my talk. But to start out with, I'm going to start with the spatial proteome profiling that we're doing, and then move more and more into the single cell work that we're doing. So when we're trying to understand where proteins are in cells, we're not using the technologies that Catherine mentioned before, but mainly microscopy instead. So this is confocal oil immersion imaging that we've automated. We have reference markers for the nucleus, microtubules, the endoplasmic reticulum, and on top of this, we add our in-house produced antibodies to target human proteins one by one. So we use these reference markers to get an overview of the cell and get an outline of where the different organelles are. And here, for example, this is CD44. We can see that it's found on the plasma membrane of the cell. So this is something that we've done for a long time and systematically. And a couple of years back, we published a paper called the Subcellular Map of the Human Proteome, where we have mapped, well, now it's nearly 14,000 proteins to 32 different cellular structures. Half of these proteins were previously never localized, not present in UniProt. We could define 14 organelle proteomes. So this is a great basic data set as a starting point for in-depth organelle studies. Since we published this paper, there's some key findings in here that I will build upon in the rest of my talk. But for example, we're not generating as many new images, but we're working hard to interpret these images because there's much more information in them. For example, right now, we're looking to identify proteins that are expressed in micronuclei and heterogeneously in micronuclei in different cell types. And we also recently published a paper where we showed that there is a new compartment, if you will, in the nucleolus, this little rim-like compartment lining the edge of the nucleolus. And we can show that it's a dynamic subcompartment of the nucleolus with a distinct proteome composition. So it has about 12% of the nucleolar proteins are there, and these proteins often relocalize to the mitotic chromosome during cell division. So we hypothesize that these nucleoli rim proteins are associated with a perinucleolar chromatin and aids in the tethering of the nucleolus to the chromatin. And we could also confirm, for example, that these proteins have high intrinsic disorder. But what is very interesting when it comes to mapping the subcellular location of all proteins is that we could show at proteome-wide scale that half of all human proteins localize to more than one compartment in our cells, like this protein. It's in the nucleus and it's on the plasma membrane. And this is, of course, from a functional perspective, very important because these multilocalizing proteins may have a context-specific function and moonlight in different parts of the cell and give rise to pleiotropic effects and just increase the complexity of the proteome from a systems perspective. So this is, I would say, one of the key reasons that we need to integrate spatial proteomics measurements if we want to build systems models of cells, if we really want to understand cellular function. And this is where, you know, my brain starts to explode when I think about multilocalizing proteins and proteoforms and so on. There's just a lot of questions. So are these different? How is this sorting achieved? Is it different proteoforms? We don't know. Are the protein populations static or dynamic in the different populations? We don't know. What are the functional consequences? Most often we don't know. So we definitely need better subcellularly resolved proteomics methods to measure this. But there are some interesting numbers out there. These numbers are a couple of years old, but still 23% of the human proteome is estimated to have moonlighting activity. I wouldn't be surprised if this number is just growing and growing. So at some point we might reach 50% or higher, who knows? And 78% of the known moonlighting proteins are actually involved in disease, and 48 are drug targets. So these are very, very important proteins to study. And I will just give you one example of such a protein and what it looks like in our images. So this is an image of cultivated cells. And you can see in green, enolase 1. It's a well-characterized moonlighting enzyme. You can see that in some cells, it's found in the nucleus. In some cells, it's in the cytosol. In some cells, it's in both. And sometimes we can also see it on the plasma membrane. So just in this small population of cells, we can see great heterogeneity in the spatial distribution of the protein. And enolase 1 has known functions in these different compartments. It's a glycolytic enzyme in the cytosol. It's a plasminogen receptor on the cell surface, and it has some kind of DNA binding tumor suppressing activity in the nucleus. And then we also see additional heterogeneity, because the drawback with antibodies when studying proteins is, of course, that we cannot multiplex everything, but it's also that we cannot really compare, due to different affinities with the antibodies, we cannot really compare the signal intensities from one protein to another, but we can compare the intensities within one experiment. So here we can say that these cells express the protein at a higher level than this cell. So there is also additional heterogeneity in terms of what we call cell-to-cell variation, which most often corresponds to temporal heterogeneity of the protein expression. And I will continue along those lines. So a couple of years back, we set out to identify all the images that show cell-to-cell heterogeneity in the protein atlas, because at least they signal that there might be some kind of dynamics going on here. So to give you some examples of what these images might look like, it can be that the proteins are only expressed in some cells, that we see a gradual variation in intensity, low expression in some cells, high expression in others, or that we see translocation. This protein is in the Golgi apparatus, but in these cells that just divided, it's in the endoplasmic reticulum. So altogether, we can show that 19% of the human proteome shows single cell or cell-to-cell variability. So this is a pretty high number. So we definitely need better single cell proteomics methods for sure, in my opinion. With this, Diana, Anthony, and Christian has worked hard to try to understand this temporal proteome, or to do temporal proteome profiling. And if we think of, we have these images and we work with antibodies, we're stuck with some kind of static assays, so to say. And there are different types of temporal dynamics in cell cultivations. We have short time scale variations, such as signaling, circadian rhythm, or the cell cycle. And then there can also be some types of transgenerational variations. But in order to dissect this temporal variability, we need either temporally resolved assays, which is hard with antibodies, or some kind of temporal indicators to generate pseudotime measurements. So for this purpose, we decided to start to try to understand which of the cell-to-cell heterogeneity, how much of the cell-to-cell heterogeneity that we observe in the cell atlas can be attributed to the cell cycle. And we started out with the easiest part, just to identify the proteins that are spatiotemporally restricted to mitotic cellular structures. So that's 230 proteins, and of which 98 were previously not known to be found in these structures. But then when it comes to the interface, we decided to work with a FUCCI cell model. So these are cells that express a red and a green fluorescent marker. So the cells are red in G1, they turn yellow, and then they turn green. So by measuring in the images these single cells, we can get this trajectory of the cell cycle, and we can fit a model to it and make a linear representation of cell cycle pseudotime. So we took these FUCCI cells and we did antibody co-staining, so what we call imaging spatial proteomics, of all the 19% of the proteome that showed cell-to-cell variability previously, and also, of course, some controls that didn't show variability. And then we also sorted the cells and did single cell sequencing. So afterwards, we could map both the RNA and the protein onto the cell cycle pseudotime. So the idea with these experiments is that if the protein is expressed in correlation to the cell cycle, we would see some type of cyclic pattern emerging with, for example, low expression in the beginning of the cell cycle and high expression later. Whereas if this variability is independent of the cell cycle, we would just see a flat line. And that's exactly what the data look like. Here I'm just showing you some positive hits. So at the top here, you see BUB1B and cyclin E1, two well-known cell cycle regulators. We can see that we can get these nice temporal profiles at the ARC level. That we seen in the very striking temporal profiles at the protein level, but flat lines at the RNA level. So we believe that these proteins, the expression level of these proteins in the cell cycle are regulated at a translational or post-translational level, most likely, since the RNA is stable. So in this study, we could identify 301 new cell cycle proteins, and most of them are varying at the protein level, but are stably expressed at the RNA level. And this is the reason that I think that they haven't been discovered before, because the cell cycle hasn't really been studied with single cell resolution at the protein level that often. And we could also see that the average temporal delay between RNA and protein on these cells that cycle at a 25-hour cycle is 8.6 hours. So that is very similar to what has been shown for the circadian cycle. We find many proteins of clinical interest, for example, potential markers of cell proliferation. We also did for some selected subset of proteins, did gene silencing to show that these new proteins, some of them also seem to be able to regulate the cell cycle. They're not only passengers of the cell cycling or the cell cycling, they actually interfere with the growth. And what is also interesting is that it's quite common with cell cycle dependent translocations, particularly to the nucleus. And I will show you one example of that. This is an enzyme, UGDH. It's involved in production of extracellular matrix, and its function is in the cytosol and outside of the cell. But it has never been described to even be localized to the nucleus, but we could show with our antibodies that it's clearly translocating to the nucleus in a cell cycle dependent manner. And here we actually GFP tagged the cells and did live cell imaging to validate this translocation. So maybe it has been shown in several clinical studies to correlate to, for example, cancer outcomes. So maybe there is another function of this enzyme in the nucleus that we don't know of yet. So this is pretty intriguing, and particularly that we see this type of translocations quite often for cytosolic enzymes. So I definitely think that we need to explore at a minimum the cytosolic and the nuclear compartments with single cell resolution and proteomics. And since this is a proteomics meeting, I thought I would take the liberty to dive a bit deeper into the details or the nitty-gritties of these proteins. So we tested many proteins in the screening, and only 26% of the proteins with variable expression, cell-to-cell variability, were shown to be correlated to the cell cycle. So the majority of the proteins that show cell-to-cell variability cannot be explained by the cell cycle. They must be explained by something else. We don't know that yet. So if we call these two groups of proteins, the cell cycle dependent CCD proteins and non-CCD proteins, we wanted to ask ourselves, so what's the difference between these two groups of proteins, and what's the similarities? So starting with the similarities, we can see that there's equal variability if we measure the variability across the cell population. So we all know that the cell cycle is a dramatic cellular effect. So all the proteins that show single cell variability not correlating to the cell cycle, they give rise to as dramatic cellular variations up and down regulation. So it's clearly strong biological fluctuations. We can also see that the variability for both cell cycle dependent and non-cell cycle dependent seems to mainly be regulated at a translational or post-translational level. We also see that these proteins have lower thermodynamic stabilities than the rest of the human proteome, and that they have higher intrinsic disorder than the rest of the human proteome. So they seem to have some kind of properties that makes them interesting, in my opinion. There's also some differences between these different groups. One is that they localize to different compartments in the cell. The cell cycle dependent proteins seem to be mainly enriched in nucleus, nucleolus, and mitotic structures, whereas the non-cell cycle dependent proteins, they're also found in the nucleolus, but then the cytosol, intermediate filament, and mitochondria. So there's some clear differences there. And we could also show that they are regulated by different families of kinases upstream. So it seems to support at least that these non-CCD proteins are involved in different cellular processes. But what remains to be understood? But if we look a bit closer at these non-cell cycle dependent variations, we can see that variations in general are very enriched for metabolic enzymes. I told you that 19% of all proteins show cell-to-cell variability. If we look at all human metabolic enzymes, the number is 40%, and we can see it across many different types of metabolism. So we've focused a bit on both cell cycle dependent, but also non-cell cycle dependent variations among metabolic enzymes. So again, just to show you one example, this is HMGCS1. It's an enzyme involved in cholesterol biosynthesis pathway, and here we've stained it with an antibody in UTOS cells. Again, we see localization to the cytosol, sometimes to the nucleus, or both, and we see this variation in expression levels as well. And for this enzyme, the known function is in the cytosol. There's no description about any function in the nucleus. So what we wanted to do is to validate this, that it's not the antibody being unspecific, but we also wanted to really make sure, can a single cell reproduce this variability, or is it something inherited? Is it something clonal? It shouldn't be. But in order to do this, we've collaborated with Manu Leonetti at the Chan Zuckerberg Biohub and used this split GFP tagging platform to tag 90 different metabolic enzymes to be able to do live cell imaging. So this is our first validation experiment. We were, of course, very pleased when we could see that if we do this in a different cell line, hex cell line with GFP tagged cells, and we take a single cell, isolated clone, and let it grow until it reaches a small population. And then we see that, yes, a single cell can reproduce all the distinct protein expression phenotypes. So this really means that this is temporal dynamics. We still don't know how it's regulated. But then, of course, at this point, we were thinking, is this cell line artifacts? Is this true? I often get that question in my talks, but then we can turn to the Protein Atlas image collection. We have so many images of tissue stainings. And here, for example, if we look in immunohistochemical images from stomach or pancreas, we can see that this protein is expressed in, these are the glandular cells of the stomach, and we can see in brown that it's strongly expressed in some cells and not strongly expressed in other cells. We can also see that sometimes it's nuclear and sometimes it's cytosolic. And in the acinar cells in pancreas, all the cells in this image are acinar cells. We can only see that it's expressed in a subset of the cells, only in the cytosol, but still only in the subset of the cells. So we kind of tend to describe this as that we do see a lot of cell-to-cell variability, cell types that we think are the same in the tissue atlas of HPA, and we can actually, that variability is reproduced in cell lines, and we can use cell lines as a model system to study this variability. And we can also see it, for example, if we look at different types of cancer, so dysregulated systems, we can still often see the cell-to-cell variability across many different types of metabolism. So these are some different proteins that show cell-to-cell variability in breast tumors. So we are working hard to answer many questions here. What's the cause and consequence of these spatiotemporal variations, particularly all the metabolic enzymes that translocate to the nucleus at rare times? What do they do there? Do they have a different moonlighting function? Are they involved in regulatory or memory imprinting functions in the cell? What's the level of cellular regulation? Is it RNA or proteins? How do intrinsic and extrinsic factors influence the single cell states? Are there oscillatory systems in the cells that we are yet unaware of? And how do metabolic and proliferative states interconnect? So sorry for flirting a lot of questions to you, but this is what we're thinking of in a daily basis. And in order to do this, we realized that we need more than single antibody stains. We need multiplexed imaging. So this is something that Sanem and Anna and Frida and others in the lab are working on to build our own panels. So we have panels to visualize cell morphology, all organelles and metabolic states and proliferative markers using the Codex technology, which is very powerful, both in tissues and in cultivated cells. And then this is a technology that I think you will hear about tomorrow from Matthias Mann. So it's really his son, Peter Horvath, that mainly developed this, but we were playing a small part of it. It's a way to start to sample cells by starting from an image and then do automated microdissection and then do deep proteomics of them. So we are now integrating Codex imaging upfront so that we can do multiplexed imaging, isolate cells that have distinct metabolic states, for example, and then do deep transcriptomic and proteomic profiling of those. So altogether, we're quite interested in what we call non-genetic cellular heterogeneity. We believe that a lot of the proteome variations that we see here is actually dynamic proteome fluctuations that is not regulated by a gene expression regulations. So we believe that spatial proteomics will play a key role in mapping cellular phenotypes, non-genetic heterogeneity, and particularly pleiotropic effects. And it might provide some of the missing links to understand cellular mechanism, particularly in these rapid dynamic transition states, or when drug resistance emerges, for example. But now I thought that since most of our work still depends on that we can interpret the images that we generate, we can generate images at a very large scale. So preferably we don't want to annotate these images manually. So I'll talk briefly about some of the tools that we've developed here and the most important question that we ask ourselves. So hour about image analysis today, but I thought it was the main question. How do we classify and quantify spatial distribution patterns? It might seem trivial. We're still working with cell lines. It's easy compared to tissues, but it's a lot of challenges. There's a great class imbalance between common and rare patterns. There's morphological differences. We work with 60 different cell lines. This is the same protein in 3 different cell lines. We have the single cell variations, 19% of the proteome, and we have the multilocalizing proteins, half of the proteome. So altogether, from a machine learning perspective, this poses quite a lot of challenges, and we've had to develop new approaches and ways to deal with the data to do this. And as you heard in the introduction, a couple of years back, we thought that we could, while we're waiting for better machine learning methods to be developed, we could make use of a citizen scientist to help us annotate these images. And this was great fun for many, many reasons. And I didn't include the video material because I thought that a lot of you would have seen it already, but maybe I should have. But in the end, you can read the paper. It was a very successful citizen science project. And I think what we showed is that non-experts, random gamers can do very high quality annotations of images, and they can really help science. And if you did design the game in a good way. So we showed a path forward to how massively multiplayer online citizen science can feed into machine learning models for scalable and improved data classification. So I myself quite convinced that we'll see more about this connection between computer games and science, and computer games and health, and computer games and education as well. So that's kind of a pet project of mine. So it's an interesting field to follow. But we were, at the end of this project, we still thought that we have really, really nice data sets in the Protein Atlas, but people really don't use our images that often to develop machine learning models. So in order to inspire people to do this, we decided to run a KAGGLE challenge. The top one was run 2 years ago, where we basically asked the KAGGLers to develop machine learning models to classify the patterns that you see in these images. And we designed a challenge and the different test and training sets and so on. And then we recently, this year, hosted a Human Protein Atlas single cell classification challenge. That was very fun because it was different, because they had to build models to go from annotations at an image level to precise single cell labels. So, and this is one of the few challenges with multi-label classification. So it's been very successful and very, very fun, again, to work with people with domain knowledge and experts in machine learning without domain knowledge. So there's a paper if you want to read more about this. But what I do want to show is we take one of the best models from the first KAGGLE competition and use it as a feature, or a subset of that model, and use it as a feature extractor. And we take all the images in the human cell atlas, which is 13,000 proteins in over 30 different cell lines. And we just extract features from these images and then project them in this UMAP. So what you can see here, all the different colors are different organelles that we assigned, manually labeled each protein with. So you can see that this model can accurately separate all these compartments, which is what it was trained to do. So that's good. But what really, really made us happy here is that it can not only distinguish fine different cytosolic punctate patterns, but it can also kind of handle these multilocalizing proteins. So for example, F here is a protein that is found in the nucleus and the nucleolus. So this is the nuclear cluster, this is the nucleolar cluster, and you can see that it's right in between. D is a protein that is found in the cytosol, the nucleus, and the plasma membrane. And again, it's right in between. So it seems like this model can be used to extract features or embed these images into a format so that we can start to integrate it with other types of data. And this is the first time we've had something that can even work with a multilocalizing protein. So we were super, super excited. And I will tell you at the end of my talk what we've done with this these possibilities, this embedding function. So we tend to think about cell function through interactions. I'm sure a lot of you agree. So I think that cellular function is, of course, underpinned by interaction. So we should describe cellular function in protein communities, that is interconnected networks of dynamic protein interactions. We're not quite there yet at the single cell level, but we can all work together to reach there. So this is just some conclusions from a recent paper from Steve Gige's lab that I thought was very interesting and a good read. So they showed that they did proteome-wide protein-protein interaction networks in several lines. The boxes are identified as the kind of cell-specific interactions. They tend to link and rewire these core complexes. So it seems like it should be very important that we can actually build this cellular model that takes this into account and that also takes this pleiotropic effect of multilocalizing proteins into account. So we teamed up with TREY IDEKER at UCSD and his graduate student Joakim and Casper in my lab and decided to build this hierarchical cell model. So we took hex cells, we've imaged them with the Protein Atlas pipeline, and then also the BioPlex PPI, protein-protein interaction data, and then we embedded it using our machine learning model and the Node-to-Vec model. And then we could translate these embedding vectors into pairwise protein distances and actually calibrate that to physical distance in nanometer using sizes of known complexes in the cell. And then we could, this is a very brief description, in a typical TREY IDEKER style, build this type of multi-scale subcellular hierarchy. And this is the hierarchy. At the top, you have the cell, and then you see the cytoplasm, the nucleus, and then it goes down. So you don't have to read all the details. What I do want to say is that all the yellow systems were known. They were present in gene ontology, but all these purple systems are actually new systems. So we have worked very hard since we built this model to validate them, and we've been able to validate very, very many of them and revealed, for example, crosstalk between cytoplasmic and mitochondrial ribosomes and identified roles for poorly characterized proteins. So we think that this type of data integration across scale, which it basically is, because confocal images has a resolution of 200 nanometers to 1 micrometer, protein-protein interactions is maybe a 5-10 nanometer distance. So we believe that music captures, in this way, it captures the pleiotropic effects of the multilocalizing proteins and give protein interactions a spatial dimension. And that's why we reveal so many new potential protein communities. So just to show you the number of systems that we would get if we would use only image data, it's very few, 2 or 3. If you use only protein-protein interaction data, we get 10 different communities, but when we combine the two, it's really more than an additive effect. So I'm very excited about this type, the way that we can work now that we can embed spatial information. And with this new KAGGLE challenge, we also have very promising models for single cell spatial embedding, and then we can just imagine what kind of data integration we can do when we have single cell spatial embedded data that we can merge with other data sets. And with the very end, I just want to say that in the Protein Atlas and my lab, we're all very strong proponents of open science, sharing data tools and models. And of course, all the data is freely and openly available in the Protein Atlas, but we also, at the talent and postdoc in my lab, Wei Ouyang, built a platform for sharing deep learning models. It's called IMJOY. So it's an open source browser-based platform for reuse and sharing of machine learning models. It's a plugin repository on GitHub. It's very convenient. It supports hybrid computing modes. You can use it in Jupyter and Bider hubs. You can go here and check out our image embedding functions or test the cell segmentation model or anything there is. And there is actually we're working on a model suite that is building on this plugin. So please check it out if you're interested. And within IMJOY, we're building more and more different plugins that we're using in our project. So they might not end up being published all the time, but they end up as plugins on IMJOY at least. So for example, this is a published interactive model trainer where we start off with a known cell segmentation model, and then the model will give you images where you can manually improve the segmentation. And by doing so, interactively within this tool, you can train a very nice and high functioning segmentation model in less than 10 minutes. So that's the end of my talk, and the outlook, I guess, from my lab is that We're working a lot to build highly multiplexed imaging assays to understand co-expression of variable proteins and crosstalk between different oscillatory cellular processes. We want to identify drivers of this non-genetic proteome heterogeneity. And what I didn't talk about is that we're also doing a lot of generative modeling to build a whole proteome subcellular map. And we want to start to include genetic variants and different types of drugs to explore spatial proteome perturbations and how that affects signaling in the cells. And before I end, I just want to make a little advertisement, if I can call it that. So I'm working at SciLife Lab, the research center in Stockholm, and just last year, we got funded to form this new spatial single cell biology platform. So it's an open research platform. It's very affordable fee for service projects, and it's mainly for Swedish researchers, but it's actually open to anyone in the world if there's time and space, and there usually is. So we offer all types of assays, spatial transcriptomics and CEDU sequencing, so Joachim Lundberg, Mats Nilsson is involved, spatial proteomics, fish technology, spatial mass spectrometry, and also single cell sequencing. So this is something you can check out if you have the need to generate data where you don't have the infrastructure in your lab. And with that, I'd like to thank my group. They're really, really amazing. It was so great to be back in the lab today after the vacation, and look forward to many more days there. And thank you all for listening. Happy to take any questions.